termination of transcription and release of the nascent RNA molecule. The elongation of the transcript may be impeded by 1. Pause sites, 2. Arrest sites or dead ends, and 3. Terminators or release sites. The pause sites induce a temporary reversible block to nucleotide addition. The arrest sites stop elongation to be resumed only in the presence of elongation factors GRE-A and GRE-B. In eukaryotes, the corresponding transcription factors is TF2S, which assist in a novel transcript cleavage reaction. The release sites leads to release of RNA and RNAP either intrinsically or upon activation by factors like NUS-A, Rho or TAU. Although the nature of DNA sequences causing transcription arrest is not known, the mechanism of transcription arrest and the roles of GRE-A and GRE-B in restoring elongation can be explained in the framework of Injuum model described earlier. The transcription arrest may result when the catalytic site of RNAP is misplaced from the 3' end of growing transcript. This itself may result if the lagging product site slipped backward on the template, bringing the catalytic site out of register. Transcript cleavage by GRE-B would create a new 3' end into the right register, thus restoring elongation. Further, at some template sites, translocation may disturb the placement of catalytic site into the right register, and GRE-A induced cleavage of short segments might restore the appropriate register to resume elongation. Thus, an elongation barrier may be overcome by RNAP through a backup and restart mechanism. Termination of RNA chains in prokaryotes occurs when RNA polymerase encounters a termination signal on DNA. When this occurs, the transcription complex dissociates, releasing the nascent RNA molecule. These DNA sequences providing termination signal are called terminators. However, sometimes termination event despite the presence of a terminator can be prevented by some factor interacting with RNA polymerase. This phenomenon is called anti-termination, and the factor responsible for this phenomenon will be called anti-terminator. It will be seen that the termination actually depends on the scrutiny of not only the DNA template, but also that of the product, that is RNA, obtained by transcription. There are two types of transcription terminations in E. coli. The termination signals found on DNA can be of two types. One, those which are recognized by a protein factor known as Rho factor, which cause Rho dependent termination, and two, those which do not need Rho factor for termination, which cause Rho independent termination. Rho independent termination of E. coli. Rho factor is a protein of molecular weight 55,000 Daltons and was isolated from E. coli. It causes termination of most mRNA molecules in vitro at relatively low ionic strength, but is not needed for termination of some of the mRNAs. Rho independent terminators contain a GC rich region followed by 6 or more AT base pairs. The nucleotide sequence of the GC rich region is such that the regions of the single stranded RNA can base pair and form hairpin like structures. This hairpin like structures forms immediately after the synthesis of the participating regions of the RNA chain and retard the movement of RNA polymerase enzyme along the DNA, causing RNA polymerase to slow down or cause a pause in transcription. Since adenine uracil base pairing is weak, requiring less energy to separate the bases than any of the other stranded base pairs, the run of uracils after the hairpin region is thought to facilitate the release of newly synthesized RNA strand from the DNA template when the hairpin structure causes RNA polymerase enzyme to pause at the site. At Rho dependent terminator, Rho protein is active as a tetramer. A model for its action proposes that Rho binds to the 5' end of nascent mRNA chain and moves along the length of mRNA. When RNA polymerase is having a pause due to hairpin structure, Rho catches up, interacts with RNA polymerase and releases the mRNA molecule from template, accompanied with the dissociation of RNA polymerase and Rho factor. Rho dependent termination of E. coli. The mechanism by which Rho dependent termination of transcription occurs is still uncertain. Rho dependent sequences are 50 to 90 base pairs long, rich in cytosine residues and largely devoid of guanines. In Rho dependent termination, the Rho protein binds to the growing RNA chain and moves from 5' to 3' along the RNA, seeming to pursue the RNA polymerase enzyme, catalyzing the synthesis of RNA chain. When RNA polymerase slows down or pauses at the Rho dependent termination sequence, Rho catches up with the polymerase enzyme and pulls the nascent RNA chain from the transcription bubble. Evidence for termination by the Rho protein is that Rho protein hydrolyzes ATP in the presence of single stranded RNA, but not in the presence of DNA or duplex RNA. Hexameric Rho, which is structurally similar to and homologous to ATP synthase, specifically bind single stranded RNA. A stretch of 72 nucleotides is bound in such a way that the RNA passes through the center of the structure. Rho protein is brought into action by sequences located in the nascent RNA that are rich in cytosine and are poor in guanine. The ATPase activity of Rho enables the protein to pull the nascent RNA chain while pursuing RNA polymerase. When Rho catches the RNA polymerase at the transcription bubble, it breaks RNA DNA hybrid helix by functioning as an RNA DNA helicase. Additional information about the action of Rho protein was obtained by adding this termination factor to an incubation mixture at various times after the initiation of RNA synthesis. RNAs with the sedimentation coefficients of 10s, 13s and 17s were obtained when Rho was added at initiation, a few seconds after initiation and 2 minutes after initiation respectively. If no Rho was added, transcription yields 23s RNA product. It is evident that the termination contains at least 3 termination sites that responds to Rho, yielding 10s, 13s and 17s, and 1 termination site that does not respond to Rho, yielding 23s RNA. Rho independent terminator differs from Rho dependent terminator in the presence of a long run of uracil residues in mRNA and adenine residues in DNA, and of a GC rich region in stem of hairpin structure formed by mRNA and AT rich structure in DNA.